Yeah, so my name is Robert Tower. I'm an assistant professor here at UT Southwestern in the Department of Surgery. Started a little over 3 years ago now. Um, Originally, I'm from Canada, and I did my bachelor's and master's there, focusing on cell biology before going over to Europe, where I did my PhD in northern Germany. And this is where I really got my kind of first taste into the world of the skeleton. I worked with bisphosphonates and whether they could be used as an imaging agent if they conjugated to a dye or as a targeting agent for chemotherapeutics, looking at things like breast cancer that are known to metastasize there. After my PhD, I ended up getting a little more involved directly in orthopedic research. And since then, I've kind of spanned the gambit on different musculoskeletal components, looking at development, aging, pathologies, and things of this nature. Looking at fractures, calvarial defect, volumetric muscle loss, heterotopic ossification, digit regeneration. So I guess I kind of dipped my toes in with cancers that metastasize there, and I've been swimming in the orthopedic pool ever since. Yeah, so I mean, after I finished over in Europe, I came in to do postdocs first at UPenn and last was at Johns Hopkins, where I worked in the lab of Tom Clemens. And I think this is really where a lot of my skills were kind of honed. And I think he also brings a really unique perspective to a lot of these stories, and taught me the value of being able to communicate complicated ideas and complicated techniques to people who are not familiar with them so that everyone can have access to that kind of information. And so I think he was really one of the main driving mentors that helped get me this faculty position. But I think along the way as well, during my time there, I spent a lot of time working with Aaron James and Mimi Samarco. And all these people have really helped me kind of catapult myself when I started my own lab. So for a long time now, I've had an interest in applying these advanced omic techniques and how they can be used to answer interesting biological questions in the skeleton. And so it started off, with microarrays and bulk seq, although I know microarrays are a little outdated. From there, it went to single cell RNA sequencing, and that led me to start to learn how to do the bioinformatic analyses myself. And so I'm not classically trained in this, but I think it's really an incredibly valuable and critical skill to kind of utilize existing data and to really address a lot of these really difficult questions that were using a lot of mouse models originally. And so I really think that these omics techniques, when applied correctly, have a lot of opportunity. And so when I first came to UT Southwestern, I wanted to keep using these omics techniques to kind of further my own research program. And one of them started with understanding the role of metabolism in the formation of heterotopic bone. And so this is an injury, in usually trauma-based growth of actual skeletal material outside of the bone. And it's commonly seen in severe burn patients, high energy blast injuries in soldiers, and total hip replacements as well. And there's not really a great treatment. And so we've looked at it transcriptionally, but metabolically, we're not really very aware of what was going on. And so we did this initial screening and we identified one metabolite, atacanate, that we ended up focusing our attention on. And the literature was saying that it was going to be in macrophages and regulating their transition from pro to anti-inflammatory. And so I thought this was really going to be an ideal kind of starter project for me to kind of deal with the hot, metabolite, to look at the crossroads between immunology and bone. When we started to dig into it, it turns out that this atacanate at the tendon injury site was actually not being made by macrophages. It was made almost exclusively by neutrophils. And so the first thing I did after that, and after doing a few confirmatory experiments, was start to read reviews about what the heck a neutrophil was. And so I went from basically no knowledge to slightly more than zero knowledge, and found that they really have a lot of unique abilities. And one of the ideas that we're chasing down now is that neutrophils can actually leave an injury site and reverse migrate back to the bone marrow where they're turned over by macrophages. And this is a normal part of their biology. So neutrophils are fairly short-lived. They'll circulate around in your body for a while, and then they'll get cleared out. And we found that this metabolite, atacanate, was made at the injury site by neutrophils, but it didn't have an anti-inflammatory kind of biological function. It didn't seem to be secreted by neutrophils to regulate other cell types in the injury environment. And we end up chasing it back. And what we're doing now is looking at the potential role of neutrophils and their production of atacanate and then reverse cycling back to actually be a way that the body can sense extremity trauma and mobilize a systemic immune response in response to potential infections or injury. And this is another kind of layer of complexity that goes beyond the secretion of cytokines, which everyone knows is important in that, but you can imagine they get rapidly diluted once they enter the bloodstream. And so this would be a really proportional response. The bigger the injury, the more you would have neutrophils making this metabolite and going back to the bone marrow and saying, we need to make more immune cells to deal with this issue. And so this project kind of took a few twists and turns, but it's really turned out to be something quite interesting that I think is worth pursuing on several different angles. Um, and along the same lines as using more of these omics techniques is we're really trying to pioneer the use of spatial transcriptomics in bone. And so when I was at Hopkins, this was the first time it had been used, first in newborn calvaria. We've used it to look at digitip regeneration. And when we came here, we started to look first at just kind of the homeostatic process in bone and bone marrow and how these cells communicate. And I've since gone on to look at um, understanding the biology of fractures and how this becomes disrupted in patients with a disease called NF1. And so we've done this now in mouse models and in human patient samples. And I really think that this technology has the ability to generate a lot of hypotheses and address a lot of questions that are kind of been lingering in the field. And I'm excited to see what we can kind of pull out from these ongoing studies. I've been attending the annual ORS meeting since I came back to the US, and I really think that it's always an exciting time to bring together all the experts in the field into one place. I think you can feel kind of a renewed sense of energy when you see, the more established labs and how they've developed these projects for decades, the new trainees coming in who are filled with hopes and dreams. And so I really think it's been a valuable tool for me to see these emerging technologies and see these emerging projects and know which way the field is shifting and really open up my lab to a lot of collaborations that I've established through meeting people at the ORS meeting. In addition to the annual meeting, I also attended the ORS musculoskeletal biology workshop, where I really think, again, it was focused in a small crowd, much more intimate. And they put a lot of emphasis on mentoring and kind of helping us walk through these late stages of postdoc, early stages of faculty, and kind of answering the questions that are hard to get a straight answer from when you're just kind of out in a public setting. So I think it has been a huge contribution to kind of a lot of the projects I'm working on and my career trajectory in general. It's kind of the nature of the field that you have to get used to rejection, right? I think I can still remember the first rejection from one of my first manuscripts. And of course, I took it very hard. This is something I spent years working on that I thought was amazing. And group of my peers said, you know what, it's maybe not as good as you think it is. And so luckily I've been rejected so many times now that it's starting to remove the sting. I don't know if that's a good thing or a bad thing, but yeah, I think it takes a lot of perseverance to kind of keep pushing through these things. And before it was manuscripts, and now it's getting into the grant funding. I hear people talk about the good old times where the pay lines were 20%, things like this, and now we're dropping down to what, 8%. So, I mean, that means 92% of the people are going to fail, right? This is not great odds. And so I think you just have to keep trying and you have to make sure that you don't get discouraged, that someone says that your story can be better. You have to take it as this, positive encouragement and do something to make your story better, rather than letting it get you down and think my work is never good enough. And so I think, I've been fairly fortunate to secure funding for the lab, but I promise you, I've been rejected far more than I've succeeded. And I think that's just kind of the nature of the business we're in. Um, but I think this also brings a lot of advantages, right? If you can, go through all this trouble and finish your postdoc and you finally start up your own lab and you have to figure out what equipment to buy and how to do an iCook and, who to hire and what projects to follow. When you finally get that success, I think it really means a lot that this is really something that you built on your own, right? This is something that you decided was an interesting topic. And honestly, sometimes it's just to understand the basic biology of the body, and sometimes it really has clinical implications that could go on to help people. And I find that really fulfilling. I think this is something that you really have to want. I think that there are better jobs out there that have better hours, that have better pay, and that'll have less stress. I think, to some extent, when you start your own lab, you are just kind of thrown into the deep end and you have to sink or swim. Um, and there'll definitely be some tough times, right? And I think especially in those first couple of years, my mentor told me that the first 3 years should actually be miserable. And so I feel like I'm right on pace to be successful because I've definitely had some misery in those years. But like I said before, I really think that compared to, something, an industry where it's, the projects are more directed and you have to have some kind of outcome that's going to be beneficial to, patients immediately, or it's going to be, shelved because they're in the business of, throughput to make drugs and make money essentially. But there's something really satisfying about, these projects we have where you think it's going to be one thing and it goes a completely different direction. And instead of saying, you know what, let's just throw that away because it's not what we expected, you chase it down and you find something that's really, I think, really interesting that maybe no one will care about, and maybe will have a huge impact in the field for years to come. And so you never know. But in the end, it's like you choose your own fate, right? You get to decide where you go and how you get there. And I don't think there's a lot of jobs out there that allow you to do that.